Thanks very much. Thanks so much for this opportunity, and it's great to be here. So, coming from Singapore, do come and visit us if you're nearby. It's a nice place to visit as well. It's always a lot warmer. It's great to be in cool weather though. Thanks. So anyway, our lab is working on cellular immunology in humans. Today I'm going to talk mostly about cancer, but we work on a lot of other diseases and applications, also just interested in immune cell diversity in general. And so with humans it's difficult because you can only get samples at particular times, and most of the time you're limited to studying immunology by looking at cells in blood. And so a way to get around this is to sort of leverage the diversity of people. They are all exposed to different things, and they have different genetics. And so when you look at the heterogeneity within people's samples and between people, you can learn about mechanistic insights. And so I think that this systems level approach to studying human immunology has been really a terrific progress. And so our take on this, though, is to try to improve this by being able to look more specifically at relevant cells. Because, you know, there's so many cells in blood, and at any given time, most of them may not be so relevant to the disease. So for instance, we want to look at cancer, and if we have only blood cells, very few of them would actually be influenced by cancer, depending on the type of cancer or really how bad it is. And so we think we can learn a lot more by focusing on relevant cells, and so that's the general thrust of things. And I think there are two major ways that we try to do this. One is by broadly profiling cells and doing comparative analysis, and try not to focus on particular cells ahead of time that or certain, look broadly and be open-minded about small changes that can happen, to look for cells that are affected by the changes, so that would be a broad perspective. And then I think an especially powerful way when it comes to looking at T cells is to focus on the antigen-specific T cells. So I'll talk about ways that we can identify antigen-specific T cells. And as I mentioned, I think this is especially important in blood, where most of the T cells are not going to be specific for whatever infection or disease you're probably interested in. So the vision that I have for my lab and in general is to leverage the specificity of T cells to develop more accurate methods for personalized medicine. And I think cancer is a really exciting topic, but other infectious diseases we're also working on, and it's also broadly applicable. And just to mention, we started a service company that can do this type of profiling, and especially looking at antigen-specific T cells. And so especially for industrial partners, if you're interested in any of these technologies, don't hesitate to get in touch with me or with them. And so the basis of this is really the antigen specificity of T cells. And so just for those of you who don't think about this all the time, this is a picture of a T cell receptor in complex with a peptide MHC. And so it's this peptide that is derived from cancer or virus or something foreign, and T cells would be selected and then for their ability to bind specifically to this complex. And so we're going to be focusing on this, and when we talk about the antigen specificity of T cells. It's a really exciting time for T cells in tumor immunology. I'm sure I don't need to mention this to this audience, but of course there's many different ways that T cells are being leveraged for cancer immunotherapy. And so it makes sense that we should look at these cells. And then of course it's in parallel, there's been a huge amount of progress, since at first the role of T cells in controlling cancer was a little bit controversial, but now quite well established that T cells can indeed control cancer, and cancers have to find ways to escape from this, and that would be immunoediting, and that comes in many different forms, such as the expression of checkpoint markers and things. And then another interesting piece of evidence with respect to what T cells are targeting when they are effective in cancer immunotherapy is this observation that cancer types that have higher rates of mutations seem to respond better when it comes to checkpoint blockade immunotherapy. And so in green here, sort of the tumor types that are more frequently responsive to checkpoint blockade as opposed to those that are less frequent, and their range of mutational burden And I'm just going to talk about, because in this talk I'm going to talk about two different types of cancer, colorectal and non-small cell lung cancer. And just what's interesting in Asia, as I'm sure a lot of you are aware, is that there's a high prevalence of EGFR mutant lung cancer. And those tumors also have quite low mutational burden as compared to the wild-type EGFR non-small lung cell. And so this paper actually did a little bit of a retrospective analysis on checkpoint blockade trials and pointed out that indeed these tumors with low mutational burden respond less well to checkpoint blockade immunotherapy. So I'll come back to that. So big questions that we have, of course, are, why do some patients respond and others not? And we talked about this neoantigen aspect, so the mutational burden. So one hypothesis, of course, is that there's a lack of antigen-specific cells in the people that don't respond. And then there's also evidence that you need to have T cells getting into the tumor. So this whole tumor hot and cold aspect in terms of hot tumors having larger numbers of T cells getting into the tumor. You could imagine also that there'd be other types of dysfunction, and that's what's driving the research into other types of checkpoint blockade strategies. And this leads to what can we do in people that don't respond. And we would like to try to find other types of antigens that T cells target to try to understand how maybe those could be boosted in addition to these what seems to be the neoantigen specific cells that are already having evidence for being important. So the topics that I'm going to talk about today is one, looking at the heterogeneity of T cells infiltrating into tumors. And then I'll spend most of the time talking about how we're looking at the antigen specificity of T cells infiltrating those tumors as well. And so we're interested in looking at T cells because then, I guess one of the ideas is that the T cell has and that was sort of the cuteness of my title, is that the T cells can see the tumor. And so if we know the T cells that can see the tumor and that they're specific for the tumor, that antigen specificity is going to influence what they become, and so what their phenotypic profiles are. And from our data, we know that T cells are extremely diverse. And when you look at T cells in blood or in tissue, there's, you can figure, it's hard to figure out how to categorize them all because there's so many different types, and there's many different ways that you can think about how they're heterogeneous. And so I think this could be powerful because by looking at the tumor-specific cells, then you can understand sort of the nature of what they're doing, and this should give you a powerful way to predict the outcomes of the patient, or better ways to treat. And so this diversity comes in many forms. One is about whether the cells have seen the antigen or not, how many functional characteristics of those cells, so can they produce various cytokines or kill or other things. We can also measure whether they've proliferated and what their abundances are. And then we're also very interested in trafficking receptors. I mentioned, T cells need to get into the tumors, they need to get to the site of infection, and so we're interested in what controls the localization of T cells between tissues and then also within tissues. And then, of course, there's many other markers that we're all interested in, such as the checkpoint markers or markers associated with other types of dysfunction. And so the nice thing about all of these markers or these types of diversities, that they can be measured using flow cytometry. So we're using flow cytometry, and with mass cytometry, which I'll introduce briefly, we can sort of look at many more different markers at the same time and probe the relationships between all these different markers. So single cell mass spectrometry or mass cytometry, also called CyTOF, is a form of flow cytometry based on heavy metals instead of fluorophores, and this allows you to stain the cells the same way you normally would with a flow cytometer, and then inject one cell at a time into an ICP-MS mass spectrometer to quantify the atomic content, and then therefore quantify the abundances of each of the tags. And this allows us to look at more than 40 different antibodies at the same time, and I'll talk about also using it to probe the antigen specificity of T cells. And so this this ability to look, you know, at many different types of, many different markers on cells. And at the time that we first started using this, the typical fluorescent flow cytometers were less than 15 channels. And so we were challenged with this whole high dimensional data thing. And my preference for trying to understand these type of data is to maintain single cell resolution. And so in this first paper, we used principal component analysis to take these 25 parameters that we measured about human CD8 positive T cells and boil them down, in this case, into 3 different dimensions. And then we colored the cells in these plots by the already known phenotypes of human T cells. And it fit in that we could see that the naive cells were more or less distinct from non-naive like T cells, and we could describe a continuum of phenotypes. And so there's a lot of parameters underlying this in terms of all the different surface markers and intracellular cytokines that T cells can produce, and so we were able to describe how all those things are related to each other. But we were always also realizing that principal component analysis doesn't do so well in terms of being able to see small subsets. We knew that there were you know, a lot of diversity in terms of cells that can make particular combinations of cytokines or express many different combinations of surface markers. And so more recently, we've been using this T-distributed stochastic neighbor embedding dimensionality reduction method. So I'm going to show these plots, and just for those of you who haven't seen this, this is very commonly used for many types of data. It does a much better job in that it plots cells that are similar to each other close by on this plot that has more or less arbitrary axes. And then what it can do is take advantage or take into account nonlinear relationships between markers. And when you have a population of cell, or each dot here is a cell, and when they're close by to one another, and they make these distinct clusters, then those cells are more or less distinct in terms of their phenotype. So I think it gives you a really nice intuitive way to see the high dimensional data and see the information that's in there. So switching over to cancer, so what we've been doing is working with clinical collaborators mostly at the National Cancer Center of Singapore. And what we're getting are patient blood, normal tissue, and tumor tissue. We dissociate the cells and freeze them so that we can come back and run them using CyTOF in large batches so that we can accurately compare the phenotypes. And we've developed panels to profile all different types of T cells. You can see we can look at gamma deltas, make cells CD4, CD8s, and we can also look at transcription factors to really profile all the different things about the T cells. And we've worked really hard to make sure that our staining is robust and accurate for each of the markers. So I'm going to further zoom in and just talk about CD8 positive T cells for the rest of the talk. And so here's an example of how we can gate on CD8 positive T cells, and then we can look at the various markers expressed by these CD8 positive T cells that are infiltrating tumors. And you can see already that many of the markers that we're interested in are robustly quantified using mass cytometry. And here are just 3 different patients. So these are separate runs of the TC, so you can't really compare between the patients in these plots. But what this plot shows is that you know, for these 3 different lung cancer patients, we've overlaid the blood in blue, and then the normal and tumor tissue in yellow and red. And you can see that the blood-derived T cells have distinct phenotypes from those that are in the tissue. And in the tissue, there's also a great deal of diversity of the CD8 positive T cells, and some overlap with the normal tissue. And that's conserved across all these 6 patients. And when you look at a single patient, now just looking at the infiltrate of CD8s into a single tumor and sort of showing this in the sort of maximum resolution to identify different subsets of CD8s in a single patient, you can do this for any patient. You can see that there's a huge degree of diversity in terms of markers that are associated with differentiation or tissue residence, and markers that are you know, thought of as co-stimulatory molecules or are things that can drive survival, and then also in terms of these checkpoint markers or markers associated with dysfunction. And so when we've had a very difficult time in that every single tumor looks quite different, and there's this huge amount of diversity between each of them. And that's sort of shown here in that, in this case, we ran a batch of samples, so 6 different patients all together. So now we can compare the phenotypes, and you can see that each of these patients also have quite diverse CD8 phenotypes, and then also each one of them is completely different. And that's true for both lung and colorectal cancer. And so this has been sort of a challenge in that we have so much heterogeneity, and we're working on trying to quantify it in a way that we can relate it to the huge number of measurements that you can make about patients to see if we can find information. We have a statistical problem in that we're measuring so many different parameters about these cells, and then there are so many different clinical parameters or other things that we're working on together with collaborators. And we're trying to increase power by going back and doing multidimensional IHC to quantify cell subsets that we think are interesting based on the CyTOF. But another strategy is to think about it in terms of the antigen specificity. And so we have all this diversity of CD8s in tumors, and so we wonder which of these populations are really tumor specific. And so we want to try to identify really the bona fide tumor specific cells and see what their characteristics are. And so to do this, we're using peptide MHC tetramers. We can make large numbers of peptide MHC tetramers using this UV cleavable exchange approach. I won't go into too much detail about that. And then we multimerize the MHC with streptavidin that we label with heavy metal tags. And this is important because the affinity for TCR and peptide MHC is low, and so you need a multimeric reagent in order to stain antigen-specific T cells. And so we use tetramers to identify T cells by their antigen specificity. We also developed a multiplexing version of this. So what we can do is label each peptide antigen specificity with a unique combination of heavy metals, and then when T cells are specific for that single peptide, they will stain only with that particular combination. And so what this allows you to do is stain very large numbers of different antigens at the same time. So for instance, if you have 10 metals that you want to dedicate to antigen specificity, and you have these barcodes that are indicative of the antigen specificity of a given peptide, and if you have a three-letter barcode, you can choose 3 metals from a list of 10 in 120 different ways. So that means that you can actually stain for 120 different antigens in a single sample. And we can go beyond that by dedicating more channels to this. It's an exponentially increasing number of epitopes that we can screen. And then the nice thing is that with the mass cytometry, we're only using some of the channels to look at the antigen specificity, and then we have remaining channels to profile the cells. So just to reiterate sort of our workflow is that we can take individual blood or tumor samples, and we stain them all in a single sample with up to hundreds of different peptide MHC tetramers to screen for, and then we also can profile those same antigen-specific cells using 20 or 30 additional antibodies that we use to stain surface or intracellular markers. And then we run them on a CyTOF, and we have software to identify the antigen-specific cells and then look at the profiles of those cells. So one of the challenges with tumors is that we don't really, it's hard to know what, there's so many possible things that T cells could be specific for. And here's a list of known antigen targets for T cells in cancer. And many of these have previously identified epitopes that we can look at, but others, there's just a huge range of possibility in terms of what T cells can target. With mutations, it's easier because there's a finite number of mutations that a given tumor can have. You can sequence that tumor, predict antigens from those mutations, and then screen all of those depending on the mutational burden of a given tumor. And so with our approach, even though we can't be exhaustive in screening for all the possible tumor antigens, we can make fewer predictions and assumptions that are required to sort of whittle this down and get down to numbers that we can look at. And then the other thing about our approach is that what we're doing is staining the cells immediately ex vivo, in this case after they've been frozen, but we can still look at the phenotypes of those cells because we don't need to expand the cells or require that they are functionally stimulated in order to profile their antigen specificity. So we think this, and I'll show why this is important when it comes to looking at the phenotypes of the cells. So we're doing this in both mouse and human, and so switching to mouse real quick, this is a paper that just came out a couple months ago in Nature Communications that shows a proof of principle for this. We used this peptide MHC tetramer screening to screen for all of the previously reported candidate neoantigens in this MCA sarcoma model, and we identified the two dominant epitopes, and then we could quickly profile these cells in all the different, in various mouse tissues, so tumor, spleen, non-draining and draining lymph node. We didn't detect any significant hits for the other epitopes that we screened. And then we can also at the same time look at the profiles of those cells. And so this is a t-SNE plot of all the possible profiles of tumor-specific cells. And so you can see that in this case, these T cells that are specific for a single epitope and infiltrating the tumor actually have a high degree of heterogeneity. There are at least two big populations for this one, and two or three or more than 3 populations for this one. These are 2 different antigens. And what we described in this paper is that actually the composition in terms of the heterogeneity of these 2 different antigen specificities are consistently different. So these 2 different epitopes expressed, neoantigen-derived epitopes that are expressed by the tumor give rise to T cells that are heterogeneous and different depending on the antigen that are being targeted. And then and this tumor responds to anti-CTLA-4 checkpoint blockade immunotherapy, and you can see that in the animals that are treated with anti-CTLA-4, the phenotypes of the T cells are changed dramatically in the tumors for both of the different antigens that we looked at. However, and then this is a plot showing the phenotypes. Again, now we're looking at a different t-SNE plot where we're also being able to look at the peripheral tissues and tumor-specific T cells, in this case in the draining lymph node. The gray cells are cells that were not detected by the tetramers, and you can see that with anti-CTLA-4, as I showed before, the phenotypes of the tumor-specific cells change considerably in the tumor. And there's also some change of the tetramer negative cells, and then in the lymph node you can see actually much less change in that we weren't able to detect a significant change in the phenotypes of the tumor-specific cells in the draining lymph node, and this is consistent with the mechanism of anti-CTLA-4 working by depleting Tregs in the tumor, and that's been described pretty well for various mouse models, including this one. So switching back to humans, again, applying our approach, we want to screen for our neoantigen-specific T cells. We're also trying to look for tumor-associated antigen-specific T cells. And what's nice about our approach is that we can include controls. So we include many different cancer-unrelated epitopes, such as EBV, CMV, and flu, or things that people haven't been exposed to as negative controls like HIV. And then we ask what are the phenotypes of these cells in the human tumors. So this is just showing our favorite example where we got a really nice hit for a neoantigen-specific T cell population. These are essentially raw data of the streptavidin tetramer staining, and you can see that we detected a large population of neoantigen-specific cells in this one colorectal cancer patient. We also detected some EBV-specific cells in that same tumor-infiltrating sample. We also confirmed both of the hits that I'm going to talk about using fluorescence flow cytometry tetramer staining. And to make a long story short, we actually screened quite a large number of candidate neoantigens across, in this case, 12 different patients to identify 2 hits of neoantigens, and we didn't detect any of the tumor-associated antigen-specific T cells. But the fortunate thing and the interesting thing is that we actually detected quite a number of hits for cancer-unrelated epitope-specific T cells, so T cells specific for viruses and stuff infiltrating these tumors. And this is a summary of the frequencies that we detected for these different antigens. So mostly EBV, CMV, and flu. Actually quite high frequencies of cells in tumors relative to the frequencies that we detected for neoantigens. So what this shows is that there are these cancer-unrelated antigen-specific cells also coming into tumors. Obviously, we were concerned about blood contamination. So these graphs show that these tumor-infiltrating T cells, this is the blood on one side and the tumor infiltrating on the right side, they actually express markers like CD69 and CD103 which are never expressed to this extent in blood. And so we think this is strong evidence that these cells are actually infiltrating the tumor, and their phenotypes are just like any other tumor infiltrating T cells. And so when we broadly compare those phenotypes, we can see that there's actually a huge range. So each of these dots is a different patient with a different epitope that we detected for these cancer-unrelated antigens in blue, and then the 2 neoantigens hits that I was talking about in red. And so there's not a lot of statistical power for the red ones, but for the blue ones we have quite a large number of samples, and you can see a lot of diversity in terms of the expression of many markers. The end of the story is that they look a lot like tumor-infiltrating cells. Many of them express PD-1, TIGIT, other markers associated with being activated. One marker that we were surprised by is the lack of expression of CD39 by these cancer-unrelated antigen-specific cells. And in all the examples that we have for neoantigen-specific cells, they all express CD39. So we think that CD39 is an interesting new marker for defining tumor-specific cells. I'm going to skip this other example for interest of time, but we also looked at an EBV-driven cancer, and these cells also, in this case, when we look at EBV-specific cells in an EBV-driven cancer, the EBV-specific cells look like tumor-specific cells in that they express CD39. So that also sort of supports our hypothesis about CD39 being a more accurate marker for tumor antigen specificity. We've also done some work to compare the profiles of these CD39 positive versus negative cells. And the bottom line for this is that the CD39 positive typically have more of the exhausted phenotype. This is also where all the proliferating cells are. So even though you call them exhausted, in many tumors, these CD39 positive cells are also Ki67 positive and proliferating. And so so then based on this hypothesis that CD39 is a good marker for tumor antigen specificity, we went back and looked at all of our data, and we noted actually that in patients, the frequencies of CD39 positive CD8s is quite variable between patients. And so in this case, we have the colorectal patients that we looked at, and you can see this huge range in terms of frequencies of these CD8 positive T cells that express CD39. And similarly in lung, we got quite a large range. And so in line with what I was talking about before, we actually see a big difference between the EGFR wild type tumors and the EGFR mutant tumors. And so the ones that have this EGFR mutant and are known to have much lower mutational burden also have a much lower frequency of CD39 expressing CD8 positive T cells. So we think this also further supports the notion that CD39 is a good marker for tumor antigen specificity. In colorectal, it's interesting that we weren't able to identify such a correlation, in that mutational burden does not correlate with the frequencies of CD39. We don't have a large number of MSI high cancer patients. MSS patients are known not to be responsive to immune checkpoint blockade immunotherapy. So this to us is quite intriguing and maybe suggests other possible antigens at play. As an overall summary, I wanted to talk about using mass cytometry to analyze T cells or immune cells in general, and then focusing on our analysis of CD8 positive T cell heterogeneity in tumors for this talk. And then I described this multiplex approach that allows us to rapidly screen and simultaneously profile antigen-specific cells, and we're seeing diversity in terms of the profiles of those tumor-specific T cells in mouse and in humans. By looking at the profiles of these antigen-specific cells and comparing the cancer-unrelated antigen-specific ones, we have developed this hypothesis that CD39 is an more accurate marker of CD8 positive T cell tumor specificity. And then we have this interesting relationship with EGFR status in non-small cell lung cancer. And so far with colorectal, we're not so sure. So I think the implications for this are important because it implies, the nice thing about CD39 is that the cells either express it or they don't, unlike PD-1 where you have different levels of expression. And so it's quite easy to quantify. And so we think that we can more accurately quantify this, and our data are showing that some patients have very few cells that express this marker, which implies that they don't have any T cells specific for the tumor actually infiltrating those tumors. But then you do see these unrelated cancer-specific cells coming in, these unrelated antigen-specific cells coming in. We think they're just coming into these inflamed conditions because that's what T cells do. But I think there's other evidence showing that in some tumors there really are a lack of tumor specificity. We think that therefore maybe this could be useful as a biomarker, and so we're exploring that. We're trying to develop IHC that can stain for this marker. It's challenging though because CD39 is often highly expressed by tumor cells. It's also highly expressed by Treg cells and B cells and other cells. So it makes this quantification using IHC quite difficult. We also think that you could potentially sort by this marker and then expand T cells that are tumor specific and use them for adoptive therapy as opposed to trying to expand all of TILs. And then we're really interested in what's happening with respect to this microsatellite stable colorectal cancer, where we don't have a good relationship, and yet we see what looks like quite high frequencies of tumor-specific cells, and they're in non-responsive tumors. And also an interesting note that we looked at from some of our data, and this had been published in another paper, that upon treatment with anti-PD-1, in some patients you see activated cells in the blood, and this has been reported. And in our case, we even looked quite early post-treatment. We can see very, very high frequencies of CD8s in the blood that are activated. And in addition, many of those cells also express CD39. So maybe it would even be an interesting marker to look at in terms of tumor specificity in the blood. It's hard to understand the mechanisms of that, but we're also looking into that. So I will stop with that. In my lab, the main people that contributed to the human work that I showed was Yannick Simoni, who's really doing all this cancer profiling, and Cheryl Lowe, who was working with him to deal with all these samples. Etienne is a bioinformatics person that was also helping out. I showed also Michael Fahling's work that was published, and he moved over to the company that we started, so he'd be happy to work with you on any other types of projects, and then a lot of contributions from other folks in the lab, and then a major contribution, especially from Daniel Tan and Ian Tan, medical oncologists at this NCCS in Singapore, and also the pathology department, sort of led by Tony Lim. We also work closely with people at the Genome Institute of Singapore, and Wei Wei Zai was the main person doing the bioinformatics for the epitope analysis. And a lot of these methods were developed during my postdoc at Stanford with Mark Davis. And then, of course, I have so many other things that we're using the same approaches for that I'd be happy to share with you offline if you're interested. So thanks very much.